So hopefully, after viewing those videos, you have a basic understanding of the role of MHC proteins and their job, which is to present epitopes, that is parts of pathogens, to other components of the immune system. So who exactly are they showing these things off to? The immediate answer is the T cells or the T lymphocytes. So if the antigens represent bad guys or parts of the bad guys, the T cells are some of the good guys. So it's their job to recognize the foreign epitopes that are being shown or shown off on the surfaces of those antigen-presenting cells. Again, these can be either phagocytes, which are the kind of professional antigen-presenting cells because they actively patrol and look for invaders, or in the case of any cell which becomes invaded by a virus, that components of that pathogen can be degraded and then presented on MHC complexes, in those cases MHC1. So make sure you understand MHC. That'll make our discussion about T cell receptors much more understandable. So the T lymphocytes circulate in the lymph, in the blood. They migrate to lymph nodes where they interact with antigen-presenting cells and B cells. They're present in large numbers in the spleen and other mucosal and some of the mucosal associated lymphatic tissues. We've brought up the Peyer's patches, for example, in the intestine. Sometimes we refer to these kind of like tonsils for your guts. So those are always sampling things that may be present in the intestines and on the lookout for potential pathogens. So the job of the T lymphocytes, they are the sole participants in cell-mediated immunity. So the job or the function of cell-mediated immunity is to recognize cells infected by pathogens, so viruses, intracellular microbes, as well as body cells that may be abnormal for some reason, producing cancer cells is a good example of that. They also interact with the humoral arm of the adaptive immune system by stimulating and regulating the various aspects of antibody-based immunity. So T cells involved in cell-mediated immunity, B cells we'll see are primarily responsible for the antibody-based immunity. Okay, so within this group of T lymphocytes, we can make distinctions between different types of lymphocytes based primarily on what sorts of jobs they do and what sorts of cell surface glycoproteins they possess. So the site of maturation of all of these, because they are T cells, is the thymus. But the ones that are directly involved in killing what I call problem cells would be the cytotoxic T lymphocytes. So we're going to abbreviate those TC. Sometimes these are referred to as CD8 cells because they contain a CD8 cell surface glycoprotein. And these stabilize interactions of the T cell receptors. And I have some slides in a second to illustrate how that works. The helper T lymphocytes are abbreviated TH. And those start out undifferentiated and then they change into either TH1 or TH2. Both of these have CD4 receptors, and their function is to secrete cytokines, interleukins, and interferons. And then finally, the regulatory or TR lymphocytes. These also have CD4 receptors, and the role of this class of cells is actually to downregulate or suppress the adaptive immune response. And you may wonder why this is important. You'll see later when we get into diseases in particular that sometimes the immune system can kind of get out of control and sometimes cause as much harm to the body as the pathogen itself. So the regulatory T lymphocytes kind of try to keep a handle on things in the immune response, specifically actually with respect to particular autoimmune diseases. So you may want to check out then the following animations, one on cell-mediated immunity, helper T cells, just an overview of how those are different. And let's see if I can advance here to the next one. Okay, so some of the T helper cells, we said, secrete cytokines, including interleukins. Just to remind you that cytokines are soluble proteins that act as intercellular signals. So cytokines, well, anything kind of small that can facilitate communication between different components of the immune system. They are secreted by various lymphocytes as well as phagocytes, and we can actually consider it the signaling network as cells, and these signals are sent back and forth. What kind of signals are they? What are they talking about? Interleukins, so inter meaning between, leukin meaning leuco or white blood cells. These are molecules which allow white blood cells to communicate with each other, and their action results, in the case of IL-2, of cloning or amplification of cytotoxic T cells in response to detection of a particular antigen. IL-4 is also very important, in this case secreted by the type 2 helper cells. Those stimulate B cells then to differentiate into plasma cells. Plasma is kind of not the greatest name, doesn't refer to just a cell in the plasma, but the plasma B cells are the ones that are mature in producing antibodies, which are then made and secreted into the blood and the lymph. So other ones include interferons, which we introduced last time as being involved not only in interrupting viral infections. Sorry for the background noise. I'm sitting in the car at the kids' taekwondo lessons. I'm trying to record this. And tumor necrosis factor, which as the name suggests, is involved in triggering processes involved in suppressing tumors, apoptosis, meaning the same thing as necrosis, death of a cell, usually programmed death. So the actual interaction between an MHC epitope complex and T cells occurs at the T cell receptor, abbreviated TCR. You'll notice that, like the MHC protein complex, it includes transmembrane regions which anchor it into the cytoplasmic membrane, as well as an epitope binding site that extends outward from the cell. So each of the cell's TCRs, or T cell receptors, have just one of these sites, an epitope binding site, and the nature of it, or what it binds to, is going to be determined by gene arrangements. In fact, there are something on the order of 100 million or so different T cell receptor types that can recognize distinct epitope MHC complexes. So now these aren't, again, recognizing epitopes that are just floating around by themselves. The only things that TCRs can interact with are epitopes that are associated or presented by an MHC protein. The T cell receptor therefore functions as a synapse between the antigen presenting cells and the T cells. I like to say, I don't know what I'm looking for, but I'll know when I see it. This means that at some point, if there's an antigen presenting cell that's displaying an MHC with an epitope, there will be some T cell that will be able to recognize it. So let's at this point go ahead and look at the initial processes that occur during the initiation of a T cell response. So again, we're talking about cell-mediated immunity in this case. And so at the periphery, which means someplace maybe in your arm or your leg, maybe a finger gets cut, and a microbe is introduced. So a dendritic cell at the periphery or at the edges of your body recognizes this is an intruder, engulfs it, processes the antigens here, and then presents them on an MHC2 molecule. The 2 isn't specified here, but you could identify that because it's got 2 transmembrane sections. So here's MHC. The dendritic cell is a type of phagocytes presenting the antigen. It then actually migrates, it travels to a nearby lymph node. And at the lymph node, and the lymph nodes are where T cells, lymphocytes interact with each other and with any filtered pathogens. But in this case, again, we've got a dendritic cell. It's processed a microbe, presenting as an antigen. So here we have a T cell that happens to be present in the lymph node, just sort of waiting around, And all of a sudden we see, well, look, there's this T cell receptor is able to recognize this same antigen. So at this point, this is an immunological synapse. That's what that's called. So again, only by virtue of the fact that that antigen that was on the MHC protein is that connection able to be made. So again, this shows this dendritic cell. This is MHC2, but T cell receptors also bind to any MHC1 molecules that are bearing antigen. A summary then of the cell-mediated immune response, beginning with phagocytosis and antigen presentation by a dendritic cell. We can see here it's not showing the processing, but the end result is some of those epitopes are loaded and presented on MHC1 molecules and others on MHC2. And you'll see that depending upon the T cell, some of those will interact with either the MHC1 or MHC2. So in this case, a T helper cell is able to interact with this MHC2, and there's cytokines secreted by the dendritic cell, interleukins, which then stimulate differentiation of that T cell into a T helper cell 1. These also secrete interleukins, IL-2, this was IL-12 here. You don't need to remember the different types of ILs, just that they are involved in communication and activation of lymphocytes. In this case, this synapse here is enlarged on the right, showing the dendritic cell with MHC1 in this case. Here's the epitope, the T cell receptor on the surface of the T cell. Remember, some of the T cells are called CD8 cells because they have the CD8 protein which stabilizes those synapses. That would be an example of a cytotoxic T cell or TC. So again, the dendritic cell at this point has already processed that antigen, is presenting those epitopes on MHC1 and 2. What again is the role of these cytokines? Well, when the body decides to mount an immune response, it really wants confirmation that this is something really, that this represents a real threat. So the fact that interleukins are being produced by the dendritic cell is letting these lymphocytes know that yes, this represents a real situation, and the T cell here, once activated, then stimulates this interleukin, which is responsible, acts as a confirmation really, to tell these T cells that it's okay to differentiate. Once identical copies of this T cell are made, those also produce a self-stimulatory interleukin cascade, and the end result is that we have lots and lots of these cytotoxic T cells. So these are activated and they are programmed to recognize the specific epitope, okay, to which they first recognize by virtue of their T cell receptor. So we said that the T cell receptors recognize a wide variety of foreign antigens, upwards in the 100 million or so range. But what prevents them from reacting with proteins or molecules that your own body makes? Clearly there has to be some protection mechanism, and this slide explains that in some detail. So let's walk you through the illustration here. And we have a stem cell, and we're in the red bone marrow, which is where these are being made. And T cells are made here. And depending upon, again, the gene rearrangements which occur, we will have T cell receptors that all have differently shaped binding sites. So only 5 are shown here, but again, these number in the hundreds of millions. At this point, the T cells are screened. So essentially, there's an evaluation process where any T cell receptor which does not bind major histocompatibility complex, this essentially renders them useless, right? So even if they can bind epitope, if they lack a receptor which enables them to interact with the MHC, they're useless and those undergo apoptosis. Okay, so these are not really even a threat with respect to autoimmunity. But then we have a case where, well, what if the cell is able to bind to MHC? Those receive then a survival signal. So these survive for the time being, but then they're screened to see if they recognize autoantigens. So their cells, they're in the thymus where these are maturing, that the T cells, the immature cytotoxic T cells are run against to make sure that there's no self-reactivity. As long as there's no self-reactivity, those are allowed to mature. So this is, or they're allowed to survive. These are the immature T cells. Those will later be activated in response to an epitope which binds specifically to the antigen binding site. If there's any cross-reactivity though, those are removed. Most of those are removed by apoptosis. A few of them, those survive, but they don't turn into cytotoxic T cells. They actually turn into more of a police force, which regulates, we mentioned briefly, the immune response to downregulate or prevent overstimulation, and those are regulatory T cells. We're not going to say a whole lot more about that just because of a lack of time, but understand that those arise from, those can actually recognize autoantigens, but they would not kill them. They would just mediate or downregulate the immune response.